a little bit about synapses. And then with the beginning of the third week, we get into the nitty-gritty of the course, which of course is the fact systems. And most of that is unfamiliar to them. And then I think that the students find the reviews a lot more useful. Today we're going to talk about neuroanatomy. And by the way, as I told you, Richard Lamb is, the old term was photographing. He's recording these sessions, and each week he'll put the disc down in the Biomed library, and there's also a set of tapes of a previous set of lectures of mine. So between the two of them, you can use those to review for short periods if you need them. We want to try to make it as pleasant for you as possible. Okay, let's talk a little bit today about how the central nervous system develops. You know, the brain is an extremely complex organ. We can tell you that. The remarkable thing is that the human brain can develop in 9 months. That doesn't mean the developmental period is finished, of course, but it does mean that all of the major systems are at least sketched out. So in 9 months, you sketch out the systems, and then you take another 25, 30 years to finish up the systems. But the most dramatic thing is that your nervous system remains plastic all your life. When you enrich your input, such as you're doing by coming to college, you actually make more connections. You know that. When you spend a nice 3 or 4 weeks on the beach, which is lovely, you get a sunburn, and you lose a lot of connections. So I very often notice that for my students who've enjoyed a summer vacation, which I'm sort of ambivalent about, I'm glad they enjoyed it, when they come back, it takes a couple of weeks to get in gear. You really have to establish new connected patterns, as strange as that seems. So the brain is a living, breathing organism. The first slide shows you in relative scale some of the patterns of development of the little brain over the first 9 months of life, starting here at about 10 weeks of age. Now, at 10 weeks, this is just a visual first survey. Don't even worry about taking notes here. This is just to give you the gestalt of what we're going to be talking about. At 10 weeks, it already looks like a brain. Sure, it looks like a little lissencephalic or smooth cortical brain, such as a mouse might have. But you have cerebral hemispheres, and you can sort of vaguely see there's a structure here that's undoubtedly cerebellum, and you have brainstem and spinal cord. And then as you go up the line, 10, 12, 15 weeks, notice that there is growth in size. The cortex, the hemispheres are becoming more complicated, and then around the 23rd, 24th week, somewhere in here, gyri, indentation, begin to develop. And now we're well on our way to the more sophisticated gyrencephalic brain that we haven't seen before. Then, of course, we go up the line, we see a tremendous change in the size of the organ. A more intensive gyrencephaly develops. And by the time the little child is at term or slightly post-term, you have a very handsome gyrencephalic brain. The remarkable thing is how often the fetal brain develops into an infant with relatively no major faults. By the way, so far as we know, no brain development ever occurs completely according to Hoyle. Not only is every one of us different genetically, but very few, perhaps none of us, completely fulfill the genetic plan. So the variation is enormous. And the things that we recognize in each other are so-called personality quirks, are attitudes, emotional, intellectual, and so forth, while partly genetically determined, are also in part the results of, you might say, minor catastrophes during development. As I say, the remarkable thing is how often development proceeds with relatively little in the way of faults or mistakes. About 2 to 3% of infants who come to term have a demonstrable neural problem that may affect their lives to some extent in the future. Of infants who spontaneously abort, in other words, of failed pregnancies, it is said that 75% of those failed pregnancies have a major neurological complication. So a spontaneous abortion, a failed pregnancy, in a way is nature's way of getting rid of a defective article. And I think in that sense we're probably very fortunate. Now let's start the process. And incidentally, the neuroembryology is covered, I think, quite well in the coloring book. I hope some of you have already had the chance to at least read the chapters. Some of you may even have colored, which I certainly hope and expect you to do. Now this is, in a sense, borrowed from the coloring book. What we have, of course, is a uterus with 2 fallopian tubes and a pair of ovaries. The ovaries are the site, as you know, of the female germ cell, or egg. It's an interesting thing that nature has given each female about 300,000 germ cells. It's a question of the 2 ovaries. 300,000. Of those, about, roughly 300 will come to term once a month as the female lives out her sexually active period. In contrast, each time the male has an ejaculation, there is something like 200 million sperm in the ejaculum, the single ejaculum of a young adult male. So in other words, the female egg, germ cell, is very, very valuable. Not that the sperm is not so valuable, but at least it's present in much, much greater numbers. And I've always wondered at this enormous disproportion, and the only conclusion I could come to is that nature knew from the beginning that no male would ever get it right. So it may not be quite that awful. But when you think of how fish would spawn, the female would lay a few eggs, you know, on the bottom of the body of water, and the male would swim over and release a cloud of sperm, and hopefully a few would find their targets. In any case, each egg will develop in a little follicle. Here we see a couple eggs developing, surrounded by follicular cells, and once a month, an egg, sometimes 2, but usually 1 egg matures and is ejected from the ovary, leaving this empty follicle, which now continues to secrete hormones for a while. The egg has no sure path into the fallopian tube, but the fallopian tube has these little fingers, or fimbria, that keep sweeping over the ovary and in a sense attract the ovum, the little unfertilized egg, into the fallopian tube. In addition to the motions of the fimbria, the fallopian tube is lined with little cilia, and its wall has smooth involuntary muscle, very much like the wall of your intestine. And so the cilia are beating this way, and the intestinal, the fallopian tube wall muscles are constricting this way, and so in a sense, this enormous fallopian tube is attracting the female egg in this direction, and if there are male sperm in the tract, they come in this way, and the cilia and the muscle beat the other way to send the sperm up this way. So I often think of the fallopian tube as a kind of little fiddler on the roof, you know, a kind of marriage broker, trying to get the sperm and the egg together. Now, most of you know that the first sperm that penetrates the female germ cell, that first sperm, immediately produces changes in the membrane of the egg such that no further sperm can penetrate. And that, of course, is appropriate. So we're getting the two haploid numbers of chromosomes, one from the male, one from the female. When they're together, we have a diploid, a full number, and then we're all set to go. Now, after fertilization, there's usually a period of about 24 hours. And then the first division occurs. And after another roughly 24 hours, a second division occurs. Here we have the beginnings of the embryo. And it's rather interesting, too, folks, that in the first division, and in the second division, and sometimes in the third division, making up what, I guess, 8 cells, for this period of time, the individual daughter cells themselves remain, and the term that we usually use is totipotent. Toti meaning total, all, and potent meaning powerful, which really means that if this little slowly aggregating embryo falls apart and the cells become separate, each cell is capable on its own of producing a total embryo. This, of course, is the genesis of identical twins, identical triplets or quads, and hopefully not too much more than that. Are any of you here part of a group of twins? Do we have any twins here? It's a very interesting relationship to watch, and it's interesting to see what it does to the parents. Okay. Progressive multiplication of the cells slowly produce, this thing is a little troublesome, slowly produce a mass of cells that we call a mulberry. Don't put that down yet. We call it a mulberry. Now you're going to find that in neuroanatomy, if you can use any language but English, you're in the money. For some reason, English is considered low class. Most usually we use French or Latin. So in this course, you're going to become expert at both Latin and French, and so you can change your major anytime you want. Instead of calling this a mulberry, then, we call it a morella, an M-O-R-U-L-A, and that morella stage continues for a short time. Here it is here, and then it suddenly begins to hollow out. Here's a cross section through it, so you can see it's become hollow. And once it hollows out this way, we call it a blastocyst. Blasto simply means early, and cyst is obviously any structure that is empty inside. So the morella becomes a blastocyst, and then we begin to have very interesting action. Inside the blastocyst, a small group of cells grows a lot more quickly. And this little inner cell mass becomes the beginnings of the embryo itself. Notice here that the inner cell mass, becoming a kind of embryonic plate, right now is made up of 2 layers. This one layer here is the so-called presumptive ectoderm layer. In other words, a layer that is presumptive means sometime in the future is going to happen. A layer that's going to form ectoderm. Some of you know the term. It's another term, for instance, for skin. Now, it's an interesting thing about the presumptive ectoderm layer. Did you know that the same cells that are responsible for your skin and for your hair and for your nails, those are the cells that also are going to develop the nervous system? sometimes ask when I talk to little kids, and I tell them about this, I say, how many of you have cats? Oh, I wish you have cats. And I say, now, have you ever noticed that the alley cat, it looks sort of ugly and has short hair and so forth, looks like he's a lot smarter than a beautiful Persian or something of that sort? And they look a little troubled, and I say, is it possible that the Persian spent so much of his ectoderm on his beautiful coat, he didn't have leftover enough of the brain? They never like that. I don't know that it's true, but it is interesting. Both the outside layers of our body and our nervous system are derived from the same embryological layer. In addition to the presumptive ectoderm, we have underneath it the presumptive endo or entoderm, E-N-D-O or E-N-T-O derm. And this is simply the layer that's going to produce all of the linings of the body, the linings of your gut and your respiratory tract and so forth. Now let's go on a little farther. This is obviously a different sketch, but it tells the same story. And here we have the presumptive ectoderm, presumptive endoderm, and now in between, a new layer begins to develop. This, of course, will be the layer in the middle known as the presumptive mesoderm or mesoderm, M-E-S-O, meaning in the middle, of course. And that's particularly important to us because out of the presumptive mesoderm, a structure right here is going to, you might say, specialize or crystallize out. And that's going to be the primitive notochord. Many of you know the term. It's sort of the first beginnings of vertebrate nervous systems were built around a longitudinal tube, a notochord. Now we have, for some time, we maintained the notochord, and it serves as a source of chemicals. We call them inducers. What do they induce? They induce changes. See, the notochord will be located right about here. And it begins to induce changes in the overlying presumptive ectoderm. And what are those changes? The ectoderm begins to thicken, make a thicker plate, a so-called neural plate, and then begins to invaginate. Let's look at that on the next slide. Here we have the we have a little ectodermal plate here. The neural plate, it has thickened. Notice that it is invaginating. It's invaginated much more deeply here, and here it completes its job of invagination, and then it actually separates off from the overlying ectoderm that's really going to become the skin of the back. And now we have, for the first time, the real basis for the nervous system. We call it the neural tube. Now your neural tube, remember the notochord is underneath here. The neural tube is going to become your whole central nervous system. The spinal cord, your brain stem and cerebellum, your cerebral hemispheres. So this is the first important step in the development of the brain. And remember, it is the neural tube, which has now become a complete tube, the neural tube, which will be the central nervous system. I just told you what that was: spinal cord, brain stem, and hemispheres. But notice what's here in blue. As the invagination was proceeding, a group of cells get pinched off right here at the point where the neural tube is almost ready to arch over. And those cells we call neural crest cells. And here the neural crest cells have sort of completely gained their independence. Then they begin to migrate down and into what will be the substance of the embryo. What does the neural crest form? The neural crest forms the entire peripheral nervous system. And the peripheral nervous system is made up of all of the nerve roots, you know, that connect your central nervous system with all the parts of your body, and all the cell ganglia and so forth that are part and parcel of this. Now, the coloring book indicates what the neural crest forms, but just so we're all in the same boat, I want to put on the board the major derivatives of the neural crest. I told you that it's going to form the peripheral nervous system. Peripheral nervous system. That includes all of the nerve roots, all of the ganglion cells. We'll learn a lot more about those. It's going to include those specialized cells that maintain the coverings, the myelin coverings of the peripheral nerves. We call those cells Schwann cells. Let's see, this thing goes on and on. The neural crest cells are also going to form pigment cells. They're going to form the medulla of your adrenals. We'll say the adrenal medulla. And they're almost certainly going to form some of the coverings of the brain. We'll learn about those on Thursday. So I'm just going to put in theta arachnoid. So this funny little group of leftover cells have an enormous responsibility on their own in what they form, not just in the nervous system, but also in other parts of the body. I have a lot of respect for them. They do their work quietly. And it's interesting, they are not so frequently involved in anomalies or miscues. So let's go back now to finish my story. Here, by the way, is an actual photograph of the just united neural tube. And these relatively few cells are the cells that make it up. We call them usually neuroblasts. There's that word that, in this case, it's a suffix, blast, meaning very primitive or early. So these are the primitive early cells which, when they replicate sufficiently, are going to form the nerve cells and the neuroglia of the entire central nervous system. And here are some of the neural crest cells breaking away and beginning to migrate down into the embryo to do their job, the one we just pointed out. Now in the next one, we're looking down on the embryo from on top. So we can see the thickening of the neural plate here. The ectoderm is thickened under the influence of the notochord. And that little notochord is going to be working away, producing all these inducers. We call this stage of the development of the early embryo, we call it the induction phase, because it's the time when the products of the notochord are helping to induce the development of this primitive tube. Just a little terminology there that you might see today. Now notice something else. At this point in the cross section, we see the neural tube has actually formed. There's fusion across the top. When we look down on the whole embryo, this is front, this is back, we say anterior and posterior. Notice that the initial area of fusion is about a half the way down. That's actually about in my high thoracic region, up in this region where the first fusion will occur to make a complete neural tube. And then it's as if I had two zippers, and I pull those zippers in opposite directions, and that gradually closes the tube until all that I have left is a little opening in front. We call this a neural pore, and more specifically, the anterior neural pore. And what do you think the one in back is called? What do you think? The posterior neural pore, absolutely. Now, this is a very critical part in the early development of the embryo as it goes on to become a fetus. And we've learned that folic acid, is very important in the closure of the neural tube, the complete closure of the neural tube. For this reason, when we're dealing with women in their childbearing period, particularly if they're trying to get pregnant, we always recommend that they take a little extra folate, folic acid, so that if indeed they become pregnant, they will not have a folate deficiency during this very important first month. You see, usually in the first 3 or 4 weeks of a brand new pregnancy, the mom isn't even aware that she's pregnant yet. That's why we don't leave it a chance to say, well, when you get pregnant, let's go to folate. It'll already be too late. So as I say, for any young woman, childbearing age, who wants to be, and is trying to get pregnant, we always recommend increased folate dosage. So I do want you to remember that, and notice, as I say, the anterior neural pore and the posterior. Now, in the normal course of things, this tube should be completely closed by the end of the 4th week. But let me show you what happens if it doesn't quite work out that way. If the posterior neural pore doesn't successfully close, we have a lesion, you might say, a fault, that's known as a split spine, but that's English, that's pretty low class. So we call it a spina bifida, which simply means a split or broken spinal cord. And there are many kinds of spina bifida. You may have one which just the surrounding membranes around the spinal cord herniate out and back, or the whole cord, you see, may herniate out with the membranes, or only half of the cord may herniate out. These are not always, but usually, surgically amenable to therapy. We can't leave it that way because the individual may turn out to become paralyzed, usually will be incontinent of urine and feces, so there are many problems. So when we can, this is repaired. Now there's a very special kind of spina bifida that you can all perhaps check out on yourself. It's known as a hidden spina bifida, but again, that's not very elegant. So we call it a spina bifida occulta, occulta meaning hidden. And what you have is nothing that's very, very obvious, but you usually have almost a microscopic little tract, a little sinus tract from the area around the roots of the spinal cord. It reaches the surface of the back, and then ordinarily you have a little patch of hair in back, just about where the cleft between your two buttocks develops. And that can be completely silent all your life. If you're curious, tonight, get yourself a mirror and see if you can see a little patch of hair back there. If you can, you might be the proud owner of a spina bifida occulta. It's rather interesting. We really became sensitized to the omnipresence, the high level of presence of spina bifida occulti When the World War II started in this country, the American government developed the most dangerous weapon in the world. It was known as the Army Jeep. And the Jeep was so dangerous that it had virtually no springs. A whole generation of young men and young women riding in these Jeeps got a terrible pounding. And that often drove small amounts of coal into the saliva up the sinus tract into the area around the cord. And they got infected areas here. And these so-called pilonidal cysts then had to have surgery, painful, infection, so forth. And so we had a whole, you might say, a subspecialty that developed early in the war known as pilonidal cyst surgery because of the spina bifida occult. So it's worth remembering this curious anomaly that occurs from lack of closure of the posterior neuropore. If we have lack of closure of the anterior neuropore, there are several things that can happen. This is one of the more dramatic and awful things. It's known, again, because of its Latin form, as anencephaly, which means A usually means without. A or an. Let's see if I can make this thing work. Anencephaly, A-N-E-N-C-E-P-H-A-L-Y, recognized cephalic or cephaly. And this simply means no brain. At the level about at the eyebrows, everything else is open, so there are no hemispheres at all. Rather a horrid kind of anomaly, and gratefully not compatible with life. And that's the result of an unclosed anterior neuropore. Now, let's go back to the neural tube. Let's just concentrate on the neural tube. It's already closed, and even as it closes, it begins to develop a series of enlargements in front. The three primary enlargements are known as the prosencephalon, pro meaning, of course, first or in front, the mes or mesencephalon, meaning the middle part of the brain, and the rhombencephalon, which means the rhomboid part of the brain. Why rhomboid? Because there is a vaguely rhombic shape to this most posterior of the three divisions. Now, folks, almost immediately, the prosencephalon is going to develop a set of Mickey Mouse ears. See them coming off here? These Mickey Mouse ears are going to become the cerebral hemispheres. So what happens is we say the prosencephalon turns into the telencephalon, or distant brain, that's this. And what's left in midline is the diencephalon, and that's all of this. So the prosencephalon segues into the telencephalon, or cerebral hemisphere, and the diencephalon, which is going to become thalamus and hypothalamus. We can put this on the board in a minute. Fortunately, the mesencephalon is not very adventuresome. It remains the mesencephalon. The rhombencephalon develops into the metencephalon, which is not labeled here for some reason, and the myelencephalon. Now let me put that on the board and show you what the derivatives are, because it's really not so hard once you get familiar with it. And it is basic to our entire knowledge of what the brain is made up of. Now, as I told you, we have prosencephalon, mesencephalon, and myelencephalon. I told you the prosencephalon is going to become telencephalon and diencephalon. The mesencephalon will gratefully remain the mesencephalon, whereas the beg your pardon, I'm sorry, the rhombencephalon, sorry about that, will become the metencephalon and the myelencephalon. Now, what do these become? Well, as we've already said, the telencephalon will become primarily cerebral cortex and basal ganglia. The diencephalon will become thalamus and hypothalamus. The mesencephalon will again remain the mesencephalon, or as it's sometimes called, the midbrain. The metencephalon will become cerebellum and pons. These are all terms you're going to get familiar with and get comfortable with. And the myelencephalon will become the medulla. All of this, as some of you may know, is brain stem. Now, the remarkable thing is that all of this is proceeding continuously into the 5th, or 6th, and 7th weeks. And very shortly, the neural tube has transformed itself into a structure that you can recognize as the early brain. It looks like a brain, very much smaller. And remember when we showed in the first slide, by the 10th week, you had a perfect little mesencephalic brain, could be a little mouse just enjoying the weather and so forth. Very, very neat. But I did want you to see these transitions. Now let's go back to the slides. This side simply shows that because the neural tube is growing so quickly, actually quicker than the little fetus in which it sits, it begins to have a number of bends or flexures. I'm not going to bother you with that. I just want you to see how it bends itself and shapes itself to the developing confinement of the developing head and neck and so forth. Now, here we see a complete fetus of about this age with all those bends in the neural tube. By the way, this funny little structure here that we also saw in the last slide, this funny little structure here, this is the beginnings, and by the way, here's a word when you go home, Thanksgiving. Use this word at home. I'm sure that you're learning something. Known as the Anlage, A-N-L-A-G-E, the Anlage. The Anlage means the beginnings of. I'm looking here at this group of students, and I see the Anlage of many brilliant physicians and researchers. You are the Anlage, the beginnings of. This, then, is the beginnings of your visual or optic system. And here you actually see the retina. See this little claw-like thing? That's early retina growing out of the brain stem, which means it tells us that our eye is indeed part of our brain. Is there a take-home message in this slide? Well, that's always a rhetorical question. When I ask a rhetorical question, the answer is always yes. You get to know that. Notice that the brain stem and now the spinal cord fill up the entire length of the little organism. Here's the developing vertebral column you can see here, and the spinal cord continues right down to the coccygeal area, the little primitive tail that, of course, is resorbed. But here is the vertebral column of an adult. Here are the vertebrae around it. And look where the spinal cord stops. It stops actually usually between the first and second lumbar. And all of this is no longer spinal cord. It's simply made up of the roots that originally came out there when the spinal cord continued all the way down here. Anybody in this class who's had a spinal tap or lumbar puncture? Anybody at all? All right, good, I'm glad you haven't. When we do a spinal tap, you know what we do? We have a long needle, done many of them. You bring usually around L2, L3, or L3, L4. You first anesthetize, then you put the needle in, you break through all of the connective tissue, you break through the dura, and suddenly you find your needle in, and fluid comes out. You're getting cerebrospinal fluid, which is why you're there in the first place. The nice part here is that you can't stick and therefore injure spinal cord. That's stopped up here. All you can do is push these roots around a bit. The person may say, ouch, got a sharp pain down the leg, doc. What's the matter with you? That's part of the course. You always hit one root, but you don't do any damage to it because they float out of the way. That's why we can do a spinal tap down here, because as the fetus develops into a child, and the child into a man or woman, now the whole body is growing in length so much faster than the central nervous system and the spinal cord, that the spinal cord, relatively speaking, shortens compared to the soma, the body around it. And that's why by the time we're a young adult, our spinal cord only goes down that far. Originally, it was way down here, and I wanted that to be clear to you. This is just a cartoon that shows how we're going from a tube, the central nervous system, the neural tube. We're going to add thickness to the walls, particularly at the cortical level. And finally, we'll end up with something like this. And all of this is due to replication of cells that are in the brain stem. This, by the way, is a wonderful three-dimensional electron, we call it a scanning electron microscopic view of the developing neural tube. Here it is. You see the individual neural tube cells, the neuroblasts. Here's the overlying ectoderm. And here are the neural crest cells that I told you about. Many of them are going to migrate down and become all these other things. These cells, probably anywhere from 20 to 30 replications of them, will eventually form the entire central nervous system. We don't really know how many cells make up the central nervous system. Now we say, roughly 100 billion, which is as many as stars as we have in our galaxy. My hunch is it's probably 200, 300, or 400 billion. And then there is, there's a pruning out, as we'll see a little later on, to say nothing of the number of glial cells. So these little cells are really active during the initial 4 to 6 months of pregnancy. It cools off after that. And then, with the exception of small clusters of very primitive or stem cells, there is very little, there is some, but there's very little further replication of cells during the life of the organism. Now this cartoon simply shows how the process of replication occurs. This is the wall of the neural tube. Here's the inner or ventricular surface where the canal is. Here's the outer surface. And here we see a cell that has it's the product of a mitotic split that just occurred off the page somewhere. Notice that it runs a little protoplasmic arm to the external limiting membrane and one here. And then it sort of floats up its own arm to this point where it adds protein, DNA, and RNA and achieves a kind of, you might say, blast cell maturity. Then it migrates down along its own little protoplasmic arms till it is close to the ventricular surface. For some reason, it is at the ventricular surface that actual replication occurs. So here you have the familiar picture, like a mitotic figure. The cells split. We now have two daughter cells, and they continue what we saw a minute ago here. And as I say, this process may continue 20, 25, or 30 times in the effort to build up a sufficient number of cells to make a mature nervous system. Now, what happens when the replicative process is over? In general, the now more mature generations of daughter cells, and many of them have now made a decision. We don't understand the decision-making process, but many of them have made a decision as to whether they're going to be neurons or neuroglia. They now, you might say en masse, begin to migrate away from that central location close to the neural tube. They migrate out to begin to form the full thickness of the central nervous system. Now, the migration itself turns out to be very interesting, especially in the cerebral hemispheres where the distances are greatest because the thickness of the hemisphere is the greatest. Some of the cells unselfishly turn themselves into little ladders. And we call those radial glial cells. Radial because their process again spans from the inner surface all the way out to the outer surface. And we often call them radial glial guide cells because all the other cells hop on board and actually, almost like little amoeba, move right on up the radial cell until they take up positions more externally. It is this, as I say, this migratory progression from inside to out that gradually forms the full thickness of the central nervous system. This is a cartoon, but it tells a story. Here is the inner layer. Here's the outer layer. Here is the radial glial guide cell. And notice these little fellows hitching a ride. And then, rather remarkably, they know, especially in the cortex, and you know the cortex, its pride and joy is the fact that it is layered or laminated. As a matter of fact, many of us believe that it is the trick of developing a laminated brain, such as characterizes the cerebral cortex. It carries the secrets of higher cognitive function, of memory, and so forth. So, migration up the radial glial guide cells to the magic getting off point, and then they get off the line. A little bit later, another population comes up. Look what they do. They migrate right through this lamina already laid down to a more external layer. And then a little later on, look what happens here. Another layer even farther out. And this gives us a take-home message. In the maturation of cerebral cortex, cortex develops in an inside-out sequence. The deep layers, layers 6 and then 5 form, and layer 4, the cells scoot through and make 4 and 3 and 2 and 1 and so forth. So do remember that inside-out sequence of migrational progression of the still primitive neurons. Once they get out here, they begin to line up, as you see, and begin to form the cortex. Now, I love the next slide. Oh, by the way, how are you all feeling? You've been sitting for 50 minutes. I have some very exciting stuff that I want to share with you. And I'm just wondering, When you look friendly, but I'm not sure that's enough. Tell you what I'm going to suggest you do. I don't want to lose you, so don't go out. But I'm going to ask everybody to stand up. I mean, let's all stand up and stretch. Little Tai Chi in place if you want to. Deep breathing.